Bom dia, pessoal. Tudo bem? Queria agradecer a presença de vocês nas conferências da Escola de Educação Física e Esporte. Hoje é um dia especial, a gente tem um convidado internacional, que é colaborador do professor Luiz Teixeira, que vai apresentá-lo daqui a pouquinho. Eu gostaria de pedir a vocês que assinassem, por favor, a lista de presença e de onde vocês são, tá bom? Vocês vão receber um e-mail com um código, que daí vocês colocando o código, vocês já têm o atestado de presença na conferência, tá ok? Daí vocês podem imprimir, tá bom? É uma satisfação muito grande a gente ver os nossos pesquisadores, os nossos colegas trazendo seus colaboradores aqui na escola e dando oportunidade para que eles mostrem um pouco do trabalho que eles conduzem em seus laboratórios aqui com vocês. Então, eu não vou me prolongar muito mais, eu vou deixar que o professor Luiz apresente o professor Jacques Duysens.
Thank you, Patricia. Welcome, everybody. First of all, I'd like to say that it's a huge pleasure and honor to receive Professor Jacques Duysens for this whole week, and now in particular for a lecture about locomotion, the dimensions, behavioral and neurophysiological dimensions of locomotion. It is a very interesting topic in motor control, and it's not an easy task to introduce Professor Duysens, because whatever I say here is insufficient, quite insufficient to provide you with the dimension of the importance of the work made by Professor Duysens. And maybe in a few words, he helped a lot. He was one of the biggest names writing the history of motor control. I think it's maybe providing all of you with the dimension of the importance of the Professor Duysens' work. And I prepared a summary for his work. He got the medical degree in Leuven in 1972. The following year, he achieved a master's degree in psychology in Leuven also. He obtained another master's degree in neurology, and later a PhD in physiology under supervision of a Dr. Person, who is a very, very important name in the development of the area of locomotor control at the University of Alberta in Canada. So there was a moving transfer from Europe to Canada in this period, and the result of this work was today a very important, a classic paper on reflex control of cat locomotion. In all these points in his education, he got the cum laude, which is a distinction, which would be with honor in all of these works during the formation. He also got a Fogart scholarship to work with Dr. Leuven, who is another reference in the area. In 1979, he returned to Europe and started research as associate at the Catholic University of Leuven. In 1992, he obtained a joint appointment as associate professor at the University of Hasselt. From 1987 to 2005, he was appointed assistant professor in the biophysics department in Nijmegen, Netherlands, and since 2000 he was appointed full professor in Nijmegen. In 2007, he was appointed as a full professor in motor control in the Catholic University of Leuven, which is his current affiliation nowadays, working in the Research Center for Movement Control and Neuroplasticity. And in 2012, he was retired with emeritus status and with a research mission to allow continuing research activities, including taking up a CNPq award to work in Brazil. About publications, he wrote 2 books, and he has over 270 PubMed publications, PubMed listed papers, with hundreds of citations in many of them. He's currently editorial board member on 3 distinctive journals in behavioral and neuroscience: experimental brain research, gait and posture, and motor control. About scientific awards, I highlight, he has many scientific awards. I highlight that he was awarded with the title of honorary member of the International Society for Posture and Gait Research due to his lifetime achievements. About the scientific interest of Professor Duysens, I could describe a long list of them, but I will let you all discover by yourselves during his talk. So, Professor Duysens, please.
Thank you very much for your very nice introduction. As pointed out, I'm from Belgium, Leuven, but I've had this visiting professorship with Sao Paulo, and the result of this work with Sao Paulo people is part of what I will be telling you about today. The topic is locomotion, but I would like to emphasize that the point will be more general in that I believe that for too long motor control has been thought as control from the cortex. And of course, if you look at our nervous system, the brain, the cortex is very impressive as compared to these other parts that we have, like the brainstem and the spinal cord. But for locomotion, I want to make the point that it is absolutely essential to learn what is happening down at this level of the spinal cord and the brainstem. And this was actually something that I discovered very early on in my career. As was pointed out, I did some work in Canada, and the first scientific work I had to do involved cats, and I had to make lesions in the motor cortex of cats. And I was extremely surprised after what I had learned in my medical career before, that this had absolutely, practically no effect on these cats. They could walk around, they seemed entirely normal. So this was a big surprise for me. How can it be that the most important part, I thought, the motor control, the motor cortex, could be eliminated and you still could have a good walking behavior? And other people have found the same thing, and this is from work from other groups that showed that actually only in very specific conditions, like when you have a cat walking on precision stepping tasks like this on these steps here, or when you have them walk over obstacles and they have to land up behind the obstacle, that are the conditions that are having when the cat has problems after a deficit, after a lesion in the motor cortex. But other than that, they looked very normal. So in this talk, I would like to start on by highlighting what we know about the spinal control of locomotion. I will then go to the supraspinal control and finally end up with something about the cortex. So one of the things that will return in this talk is a general theme, and that is that what I found over this many years is that you have to look at evolution to understand locomotion and motor control in general. And in this case, evolution is not really an engineer, but it's a tinkerer. And what does it mean? Well, an engineer works in this way that he builds new things. But the tinkerer is also building new things, but he also uses old materials and old methods to build these new things. And evolution works more like a tinkerer than an engineer. That will be the point. And I will start with an example that's not from motor control, that's not from locomotion, but it's from the visual system. In the visual system, it's known that the primitive animals, like the frog, for example, has a very primitive visual system in the tectum, but it's a very efficient visual system because it allows the frog to catch flies when the image of the fly is projected on this tectum. Of course, then in evolution we went further on to become a primate. Human is also a primate. And for primates, especially the early primates, it was more important to find good fruits, ripe fruits, than it was to catch flies. And so they needed another visual system, which is shown over here, with the visual occipital cortex, having areas where they could, for example, do an analysis of the color to see what of these fruits are ripe fruits. So it's a very different system as what is known for the frog. And we end up with a brain that is very similar to this type of brain of the cat and the monkey, and the monkey and the rats, which contains now these two parts. It has this old-fashioned system here, the tectum from the frog, but it also has the newer system from the primates. And these two systems work very nicely together. So you have both old and new, and this is an example of the evolution being the tinkerer. I come now back to the topic of today, which is locomotion. And in locomotion, everybody is interested, of course, how is it possible to generate these activations in these various muscles during gaits in this nice sequence as shown here. And this happens during the step cycle, which consists of a stance phase and a swing phase. And so the first question is to try to understand how these parts of the step cycle are generated. And this is starting here in this slide with explaining what happens in this period here when you flex the limb. When you flex the limb in the swing phase, you basically flex the limb in all the different joints, in the hip, in the knee, in the ankle, there is flexion. So you need to have a spinal control that provides this flexion in all the different joints. If the evolution would have worked like an engineer, it would probably have tried to build a new model to have connections to all the various flexes in the limb and to control that by the spinal cord. But that is not what happens. What in reality has happened is that evolution has thought we don't have to invent anything new because we do already have the circuitry that's necessary for flexing the whole limb. And it's very simply the flexion reflex. When you have painful stimulus, like for example the nail that's shown here, you will flex the whole limb to protect your foot. And that circuitry can be used also for gait, because that's also the type of muscles that you want to activate in the swing phase, all flexors. So this actually was the first idea that came along, that is that flexion can be generated by the flexion reflex circuitry. And the same is true for the extension part. In the extensor phase, when you land the foot, you extend the whole limb, and there also you need extension in the various joints, in the hip, in the knee, in the ankle. And again, it's again evolution thinking about old solutions and to use that, because also it is known that there is circuitry in the spinal cord that can generate an extension of the whole limb. When you have an experimental animal like shown here, you put it down on the floor, then it will extend the limb as soon as there is contact with the feet. So there is a supporting reflex, extensor thrust as it's called. This is also true in humans, in newborn, you can have the same reflex, and when you unload the baby from the surface, you will see that there will be a flexion reflex. So you have these various reflexes are already available from evolution. So really basically, if you want to generate a step cycle, what you should do is to combine these reflexes. You should combine the circuitry that provides the flexion with the extensor center that generates the extensor activity. And you have to find a way to have this activated in the sequence. people a long time ago, this is from the turn of the century in 1905, was already the idea from Philipson that this could happen if we had a series of reflexes, if these different centers were activated in a sequence. And so you would have a basic series where you touch down the extensor thrust, then you would have the unloading and flexor reflex, and to end you would have touch down again, and so you would cycle around these various reflexes and these reflex pathways. An easy way to test this is by having an experiment that was done a long time ago, where people have tried to eliminate these reflexes by cutting down, for example, in this area here, where on the dorsal side of the spinal cord you have the input to the spinal cord, the afferents are needed for the reflexes. So if you cut down there and you would eliminate locomotion, then you would have proven that it is a series of reflexes. But the outcome of the reflex study was very different. The outcome of the experiment showed that even without the reflexes, the spinal cap, without these afferents, can walk. So this is shown from, these are the original data from Graham Brown, and it's very old data, but I think they're still relevant because nowadays there are still people believing that reflexes can explain the whole process of locomotion. But instead, Graham Brown showed a long time ago that this cannot be the case because you have, without reflexes, you have these stepping movements. And so he introduced the idea that in the spinal cord there is something called a central pattern generator. That is circuits that are acting as a generator of locomotor activity. So the central generator was thought to be originally to look like shown here, with these two centers, flexor and extensor centers, and they were coupled so that you could generate alternatingly the flexor activity and the extensor activity. And they called this center a central pattern generator with these two parts. And if you look at this center, of course the most essential part is the fact that you do have mutual inhibition between the, sorry, I'm going to put fast here, between these two centers, flexor and extensor center. And originally it looked as if the central pattern generator was a symmetric pattern generator. And it's only later on that people have realized that this may be too simple. It's not maybe not a symmetrical system. And one obvious point to raise is the fact that locomotion and the gait cycle is not symmetric. If you look at the differences in speeds and the step cycle, sorry, over here, you see there's a big difference between the swing phase and the stance phase as you increase the speed. What you see is that the swing phase is rather constant, while the stance phase decreases in duration. So you shorten the duration of the stance phase, but the swing phase is relatively constant. So there is a basic asymmetry in the step cycle already, and in some experiments, these are all data again, but experimental data showed that there may be some asymmetry in the system. And this is the type of experiments that I'm talking about. These were experiments done on animals like rats, cats, but also dogs. Having these animals walk on a treadmill, and in this case you see that the animal was walking on 3 legs, and one leg was supported and could be used for experimental manipulations like stretching muscles and so on. But the basic observation is that even on these 3 legs, the cats were able to walk. And you see this on the right. This is typical recording, alternating flexor and extensor activity, flexor, extensor activity. But the point is that in these preparations, sometimes it didn't work so well, and you had situations where all of a sudden there could be some burst missing on the extensor side, as is shown here. But the interesting point then is that in that case, the flexor burst still continues. And this is very different from other situations where it could happen that the flexor center would now fail and there would be no flexor activity here. In that case, always there was also an omission, a deletion of the extensor activity. So this points out the fact that really it is the flexor center and not the extensor center that determines the rhythmicity of locomotion in the spinal cord. So you need some input to this flexor center to make it rhythmically active. And this clock, if you like, could be a new clock, or it could be some old clock. As a thinker would think about, he would try to find old clocks. And in evolution, there are old clocks. In evolution, we stem from primitive fish and from eels, like the lamprey, for example. And these animals have no legs, but they do have rhythmic motor activity. And this rhythmic motor activity is provided by having flexion on one side and relaxation on the other side, alternating all the time to make these animals swim. So they have a clock, if you like, to provide this alternation between the two sides. Now in evolution, we then came from a situation where these fish, the swimming fish, would then at one point starting to grow limbs, as shown here for the axolotl, or some of these fish would come on land and they would develop some type of legs instead of the fins. And now the question is, how are we going to regulate the stepping of these limbs once they are on land? And this study by Griller has shown quite nicely that if you look in the circuitry that is available for quadrupedal animals like the rat, the cat, mouse, and so on, and you compare that to these primitive circuits that cause the swimming in the lamprey, you find many similarities. So the basic idea is that actually in evolution there was a rhythm generator already in the fish that was taken over in evolution by the more evolved animals like the ones that I described, the cat and humans as well. So the question then arises, do we in that case the same also a rhythm generator for the extensor part of the central generator? And the answer is no. And the reason is that the extensor part is much more connected to the outer world, to the interaction with the environment than the flexor part. During the flexion phase, there is usually no contact with the ground, and there is of course not much input, sensory input. But it's very different in the extensor phase. When you put the foot down, you have loading of the limb, and you will have cutaneous feedback from the foot. You will also have load receptors in the muscles that detect that the muscles are contracting. And these activities, they feed back on the extensor part. So during gait, during the stance phase, you reinforce the activity in this extensor part, and at the same time you inhibit the flexors. Until at the end of the stance phase, the limb is no longer loaded, the flexor part is no longer inhibited, and you can have the onset of the swing phase. This was also older work that we've done in this PhD work in Canada. This is from that work again, showing that you can have a contraction in an extensor muscle, as shown here, contraction of the muscle, which provides afferent activity in the Golgi tendon organs, which register the force in the muscle. And these Golgi tendon organs are situated at the transition from the muscle to the tendon. And during gait, you can do the same experiment. You can activate, you can make the extensor muscle contract. And what you see is that when you do that experiment, you see that there's more contraction in the extensors. That's the positive feedback, if you like, the reinforcing reflex. But at the same time, you inhibit the activity in the flexors. So this is a reflex that promotes the extensor activity in the stance phase, and by unloading it can generate flexion. Now we come to a topic that of course will interest most of the people around here, and that is, are we sure that we can that this story is also valid for the human? Because the human, of course, has a different gait. It's a bipedal gait and not quadrupedal. And so the question is whether CPGs are also used by the humans. We think we can answer that, and actually one of the answers is already shown in this slide. A newborn is already able to walk, to walk, at least to make steps when it's put on the surface, like it's shown here. And that is, although there are no connections, no right connections between the brain and the spinal cord. So the spinal cord, in this case, has to do the job of making these steps. So yes, This is one of the arguments showing that there may be human CPG. A long time ago, in '98, we used this and other examples to make the point that we believe that there is also a CPG in humans, and I'm glad that I can show you how many times this was cited over the years, and it's probably a reflection that people start to believe that this is indeed the case. There was also a study showing or arguing that there is no CPG in humans, and this study is cited a lot less. It's no proof, but at least it shows you that more and more people are believing in the CPG for the human. This is one of the people that believes in the CPG in humans. It's from work from Ivanenko. And what Ivanenko has been doing is he has looked at the EMGs, many EMGs of many muscles in different stages of human evolution, from babies to old people. And he then has a method to project that activity to the spinal cord to see which part of the spinal cords are active at a given point. And he came up with this type of figure showing that there are two levels, the lumbar level and the sacral level, where you find alternating activity in parts of the spinal cord. That's of course exactly what you would expect for a CPG, which has to activate flexors and extensors cyclically, rhythmically, and that would be perfect for a CPG. This figure and this data, along with some other new recent findings in the rat, has been the basis of our proposal that we made in this new article that just came out a week ago with Professor Forner Cordero, working in the robotics department. We propose now that this human CPG looks a bit like shown here. And you recognize already a few of these items. You recognize the fact that there is this two centers. There is a swing center and a stand center, so that's providing flexor activity or extensor activity. Sometimes you have biarticular muscles like this here, which receives input from both. And what is essential is that the swing generator or the flexor generator has this activity from the rhythm generator, from the clock if you like. There are these two levels, lumbar level and the sacral level, and it's known from literature that the lumbar one is the more important one. It's the more important part of the total generator system. There's also another point of interest here, that is this point here, that we have labeled V2B neurons that provide this mutual inhibition between the flexors and the extensors. And these neurons are identified, they are known, and they are found in different species. And this is important because it shows that the power of this new technique in neuroscience, which is called the optogenetics. And in this previous slide that I showed, we have another example like this here, HP9. It's another example of a neuron that we know exactly what type of neuron it is. It is part of the rhythm generating circuitry. So we start now in neuroscience to be able to identify the different neurons involved in the CPG. So I think it is essential for you to understand a little bit more about this technique. And let me just introduce it to you. It's a technique that uses light to activate neurons. And the way this works is that in optogenetics they use proteins called opsins, which are sensitive to light, which can have fluorescence. And these proteins and the gens for these proteins are introduced in viruses that can be introduced in particular areas in the brain. And these opsins, these proteins, can be made sensitive for a particular type of transmitter. So it can be acetylcholine, it can be GABA, it can be glutamate, and so on. And so it's possible nowadays to activate specific circuits in the brain using a given transmitter. That's optogenetics. So this is another slide illustrating that it's a light-sensitive protein, the opsins from algae, used to introduce certain viruses in the brain in certain particular areas. So we know now some parts of the circuitry, And we also know some characteristics of the CPG that is important, namely the fact that the CPG not only has the task to control the muscles in this sequence, in this nice sequence during the gait cycle, but it has another function, and it has to control the input. And this is shown on this side here. What you see is that the stance generator receives this input from the load receptors, as I indicated before, so that's this positive reinforcing feedback. But to do so, it has to, at the same time, suppress the inhibitory pathway. So these pathways have two effects. They can be inhibitory, they can be excitatory. And so the central pattern generator has the function of being a sort of police that says, no, we don't want this input, we need the other input. In this case, it inhibits the suppressive part, and it allows the reinforcing feedback. So this is the general idea, that the central nervous system gives commands to motor neurons and to muscles, and this has effects on the environment. So there is feedback, and that feedback can go in the same direction, but it can go in the other direction as well. And in that case, the central nervous system has to have a parallel signal to control the feedback from muscles and from cutaneous afferents and so on. So that's an essential feature of this new model for the human CPG. Another one is shown here to the left for the swing generator, the flexor burst generator, showing that the flexor burst generator receives input from 1A afferents. These are the afferents from the spindles that are sensitive to stretch of the muscles. And this is the long story, but just to make that long story short, we know from experimental work, especially on cats, that the stretch of this muscle here, which is the iliopsoas, this muscle here, the stretch of that muscle facilitates the onset of the swing phase. So there is also facilitation by afferent input, by input from the stretch receptors. That facilitates the onset of flexion in the swing phase. Now, so far we have been discussing the CPG as it is present here at the lumbar and the sacral segments of the spinal cords. But of course, the spinal cord also contains parts that control the arm. And we are coming from quadrupedal ancestry, like the rats and the mouse. And it is known, of course, that the forelimbs are controlled in these animals by CPGs. So people are starting to suspect that indeed in the human spinal cord there is also a presence of CPGs to control cyclic arm movements, like you're swinging your arms during gait. And this is important because there can be also interesting connections between these two levels, between the arms and between the legs. And we've done some work, I will briefly describe this work. It was inspired by the fact that people have claimed that this can be used in rehabilitation, because in rehabilitation, in some cases, the movements of the legs on a treadmill training can be facilitated by having at the same time movements of the arms. And so we wanted to investigate that, and this is the setup we used. It's not a treadmill, but it's a recumbent stepper. But we thought what we could investigate is whether the movements that we make with the arms, with these devices here that are not connected to the main recumbent stepper, if you have these movements, can you prove that the leg movements are facilitated? That was the experiment. And these are the results. When you add these arm movements, that's the red signal over here, you see that the EMGs in the legs, in some of the muscles, it's clearly improved and higher activity in some of these muscles. So this was in a normal subject, a student, but we also did some work on patients, spinal cord injured patients, with basically the similar result. That is that in some of the muscles you get improved activity in the leg muscles when you have at the same time rhythmic activation of the arms. So this for the spinal cord, we have the CPGs at the leg level, we have them at the arm level, they interconnect and they facilitate each other. Let me now briefly say something about the supraspinal control of the locomotion. And when we talk about supraspinal, the first we encounter, of course, is here the brainstem and the cerebellum. And the idea is that the CPGs need to be controlled because, of course, the CPG can only produce rhythmic flexion, extension, but of course in many cases that's not enough because you not only need gait, you need a directed gait. You want to go forward, you want to go backwards, go sideways, and you want to have a control of the speed of this gait. So you need supraspinal control. Again, this idea could lead to some new system, or it could be using older systems. And in this case, it looks as if the engineering evolution took over, because there is indeed a system known that starts all the way in the visual cortex. Let's say that I see the door there, I want to go to the door. I send a command to the parietal cortex, and the parietal cortex is information where I am in this room at this moment, and I can calculate how I can go to that door by using the motor cortex, sending a command all the way to the CPG. So that system does exist, it's a direct system. Problem is that it is a system that is probably only useful for very slow movement, because it's like when you go on an ice surface, you make every step consciously and very deliberate. But that's not very handy in most situations, because most situations of walking are depicted like here. You want to be able to walk in the streets of Sao Paulo while you're looking at your cell phone and at the same time phoning to your friend. How can you do that? That's not using the system that I just described, because that would mean that you could only very slowly move over the streets, and that would not help. So there must be another system, and there is, fortunately for us. And again, there we go to the tinkerer idea, because we need some sort of system that can automatically produce gait while you do other things like texting. So we come again to this idea that evolution has used older systems. So it pays to look back at these older systems, and in this case, this is again the lamprey that Grilmer has described so well. And it shows down here what we already described, that you have the CPG with the sensory inputs, but it also describes the supraspinal control. And the supraspinal control is by means of the basal ganglia and some intermediate structures like the MLR, this is the mesencephalic locomotor region. So basically, this is the structure that you need to control the CPG. It's needed for the lamprey fish, but it's also needed in mouse, cat, and humans. Central part is this intermediate stage, which is called the mesencephalic locomotor region. And again, it's very interesting to see that in recent times there have been discoveries about this system, the MLR. The MLR is shown centrally here. And you see that on top of this here, on the left side, you have these black areas. These are all parts of the basal ganglia, the putamen, the globus pallidus, the substantia nigra, and so on. And they are all providing inputs to this MLR and making an automated gait possible. So this is the basal ganglia circuits, and it projects to this structure that I call the mesencephalic locomotor region. This region is extremely powerful for gait, and in fact this is the sort of structure that can be stimulated electrically, as is shown here, and make a mouse walk on this rolling ball. In fact, I've done some experiments myself with animals having an electrode in this part of the brain, and it's absolutely spectacular because the animal is motionless before you stimulate. You put in some electrical current, as shown here, and you have the animal walk. And you put more current, higher current, and it will walk faster. So it's like a machine that you can regulate. So it's extremely potent, but I must admit that in some of the experiments that we also did with the MLR stimulation, it not always worked. There were also times that you could not have the animals walk. And this was a mystery at the time that we did these experiments, but it's now clearer how this comes because, again, this technique of the optogenetics. With the optogenetics, it was discovered in the last years now that there are actually several locomotor systems. What I call here an old system, the basal ganglia system, but there is on top of that a very old system, and now let me illustrate that point. Let me first make sure that I convince you that it is useful to listen to this, what is happening in the rat with these centers, the mesencephalic locomotor region, which consists of two parts, the cuneiform nucleus and the PPN, the pedunculopontine nucleus, because these structures can also be found in the human. In the human, you have also exactly these two centers, the cuneiform nucleus and the PPN. And the PPN is in fact used in pathology, Parkinson's disease, to be stimulated for Parkinson patients that have trouble starting to walk. So it is interesting then to learn more about this mesencephalic locomotor region, and you can do that by this technique, this technique of the optogenetic stimulation. And with this technique, it was shown that if you now, for example, have light on the specific neurons, the glutaminergic neurons in the cuneiform nucleus, then you can have this type of behavior. You can have a rat or a mouse that you can activate with an air puff, and it will run away as fast as possible. That's a sort of a survival run, if you like. On the other hand, if you have the same type of neurons, also glutaminergic neurons, but in another structure of the MLR, the pedunculopontine nucleus, the result is very different, because then you have that the animal starts to explore its cage. It goes to all the different corners and it's exploring with a slow walk all the different parts of the cage. So that's a different type of locomotion, and so this brings out the idea that in fact we may have several locomotor systems depending on the task that is needed. Do you have to escape from danger, or do you have to explore your cage? So this is now what people believe that is happening, that there is indeed this old, very old system that starts here with the amygdala, which is a part of the brain that is very important in signaling when there is a dangerous situation. It will elicit activity in these various structures to the MLR, and it will make the animal run away, as shown here. That's what is sometimes called the emotional locomotor system. And it's different from the cognitive locomotor system, which I described already, the one that starts over here. It can go either directly to the CPG or can go over the basal ganglia to the MLR. So that's a system that can give you automated gait. You can automatically walk around in this environment of the cage, as this mouse is doing here. So the basic idea is that the same structure, the MLR, really is part of two different systems. So evolution again as a tinkerer. So let me sum up what we have discussed so far. Reflexes are very useful. I started by telling you that because they allow the interaction with the environment. But the most important one is probably the load feedback during the stance phase, because that is important as soon as you use gait to work against gravity, as you do in quadrupedal and in bipedal gait. These reflexes should be flexible, they should be adaptable, you should have this reinforcing feedback during the stance phase and not during the swing phase, or inversely, in the stance phase you do not want all of a sudden to have a flexion reflex because you are supporting your body, so you have to have a means to control these various reflexes. There's these phase-dependent activations that I explained to you. And it's not enough to have these reflexes, because you also need what is called a central pattern generator with a flexor and an extensor center. And the flexor center and not the extensor center needs input from a rhythm generator. And the leg CPG is not different from the arm CPG, but they are influencing each other. Specifically, the arm CPG is probably helping the leg CPG. And the CPG, both of the arm and the leg, is controlled by supraspinal structures like the brainstem structures that I described, the MLR. So let me then go to the final part of the talk, when I want to say something about the cortex, because I started out by saying that this beautiful structure that you see here, which is the cortex with the corticospinal tract, ventral and lateral corticospinal tract, that it is only doing relatively little in the locomotor control. Only when you have a very slow, cautious gait it may be important. It's still important to see what the function could be in some particular situations, and as I described, there are some situations known in scat literature, for example, showing that some of these activities are not possible without the motor cortex, when you have to make very precise stepping movements and when you have to step over obstacles. So what we've decided to do in our laboratory in Leuven is to take some of these conditions and look at what happens when humans are performing these. And we will start by having an experiment that comes close to this one here, making precise steps in the lab on the treadmill. And this is how we did it. We had people walk on a treadmill, and we had them walk on stepping stones. And the difficulty was that at some point we could change the position of one of these stepping stones. We could shift it sideways, we could shift it back, we could shift it forward. And in that case, the subjects were obliged to adjust the step to make a precise step in this new area indicated by light. This is some of the data from that experiment. Of course, it is important to have sufficient time to be able to do this task. When you have very little time to react, it's very difficult to do, and that is what you see here. Normally, you should end here, and very few trials were correct. But if you allow a lot more time, you see that all the trials are in the correct space. So the precision movement depends very much on how much time you allow. And there's also a difference for the direction of the gait. If you force people to make a long step, to make a more anterior step, it's relatively easier than when you force them to make a back step, a short step, or a side step. So there's a difference also in the direction. In the forward direction, you actually prolong what you already were planning. You were planning a step, and you have to just prolong it a little bit, and that is easier. We also did some experiments that simulate what is shown here, stepping over an obstacle. And again, we used this setup with the lights projected on the treadmill. This video shows an example. You see that there is this purple illuminated spot, and the task of the subject was to avoid that, as was just happening now. So instead of making a step on this colored surface, they should make another step. They could choose to make a shorter step or longer step, but they should avoid stepping on the purple surface. So in this case, they had to make an adjustment, as is shown here. Instead of stepping on the colored surface, they could make a long step, as is shown here. And the difficulty gradation was made by the time available. We have also combined this sort of task with what is called a dual task. And a dual task is a task where you know that the cortex is involved. This could be a task where, for example, you would hear the voice say hi, and it could be said as hi, or it could be said as hi. And when it is hi, it's of course a conflict between what you hear, the sound, the tone, and the text. So it's not congruent. And it's difficult for the subject to then say the correct answer, because they had to say what tone it was. So they should say low, although the word is high. And that's called incongruent. So we did this test first without the dual task, and this is some of the results. The number of incorrect responses made to this is the dual tasking with young subjects versus old subjects. And what it shows is that when you have the incongruent, or even the incongruent, that's these two here, the young adults can do it quite well. So in other words, they have minimal cortex needed to walk. They can do even a difficult dual task, and still make no mistakes in the dual task. So they had correct identification of the high or the low voice. But it was very different when we now did the same experiment in the elderly people, because they did fine as long as the sound was the same as the text, but when the two were different, you saw that they made a lot of mistakes. And they also made more errors in the obstacle avoidance, so they could not avoid the obstacle. That is shown here. So they were, depending on the time that is available, if there was not much time left to change the direction in a short time here, then you would see that the elderly people would have a problem. In fact, we could not test the very most difficult situation, but even if you look here for the same situation, like 500 milliseconds, you see that in red that the elderly made more failures also when trying to avoid this obstacle, in this case, this colored surface. We were interested to make it even more difficult. You see that young people do not always achieve it for the most difficult situation when you have very little time to react, to change your direction of where you would place the foot. And we wanted to know what is, in the most extreme conditions, how young adults could achieve even more challenging conditions. And this is shown in the next condition. This is an experiment we did together with the group in Amsterdam. They had a stumbling setup. You can see here that there is a plate coming out of the ground at a given point in the step cycle, making these people stumble. And so we were interested to see whether people would be able to make an adjusted step after they stumble. So you put the foot down. As you see, these people are always placing, or most of the time placing their feet at the same spot here. And so we made that spot the forbidden spot and made it so difficult that they should avoid stepping on the place where they normally would land. So this is the setup. This is where the obstacle could appear, and there could be here a projection of a forbidden zone, like the colored area that I just showed you before. They should avoid stepping on that area. So this is the lab again with the projector having a light somewhere here, I'll show you. And so the normal area of landing is shown here, and they should avoid stepping on it, for example, by making a shorter step as shown here outside this area. So this would be where they would not land normally, but if they would do this during the experiment, this was an error, because they should land outside this forbidden area. This will show you how the experiment worked. You see that there was a stumbling again, and there was immediately a projection of this forbidden area, this lighted area, and they had to avoid stepping on it. And the question was whether they would be able to do it. And this is the result. These are the number of avoidance success rates, so how many times did they achieve doing it. And you see that in a sizable number, the young people were able to do it even for the first trial. But interesting, I think, for this group also in physical education here, it's interesting to see what the effect is of learning. When you repeat the experiment, you can see that there is a significant improvement in the success rate. So they were better and better after some training. They can learn to trip successfully. Now I come to the last part. I want to tell you a little bit more about learning, because we also had a question whether not only whether you can trip successfully or learn to trip successfully, but also to fall successfully, because sometimes you trip and you cannot avoid to fall. So it's important to learn to fall properly. And we believe it's possible to learn that because in our stumbling experiments, we had looked at the EMG muscles. And for example, this is the biceps femoris active during the stance phase and in the beginning of the swing phase, shown here. And this muscle is also very active in the responses to stumbling, to tripping. And this is the response that you see in the young subjects. It's a very early response. It's closer to 60 or 70 milliseconds than 100 milliseconds. So it cannot be a cortical response. So this is a very early response, which is stronger in the young than in the older subjects. But if you look at the later stages of the response here, it's not so different between elderly people and young people. And this late activity, it's well beyond 150 milliseconds. That could be a voluntary activity. So that means that after you stumble, you have first a reaction that you can hardly control yourself. But there is also later activity where you can voluntarily do something or not do something. And this is the reason why we think we could train these people, elderly people as well as young people, to try to avoid falling incorrectly. And of course, the best way to do that, everybody knows here, is that to use this technique from judo, to have a fall technique, to break your impact so that you're not having the impact on one particular spot like your wrist, but that you spread the impact over the whole body, and that you spread it also in time. That's the principle of the judo fall. And so some years ago we started an exercise program in the elderly to try to learn them how to fall correctly. And in the Netherlands, I'm proud to say there are quite a few physiotherapist practices where they have this training program to try to learn the elderly to fall properly and safely. This is an example of how it is done. There's a mattress, and people are trained by a physical therapist to learn to fall in the appropriate way. So this sums up the second part of the talk, saying that I was pointing out the fact that in these responses, like for example in the tripping responses, some of these responses are very fast, less than 150 milliseconds, and we should not forget that the brainstem may be very important in these responses. It's certainly too short to be a reaction that can go all the way over this conscious part of the cortex. You can learn some of these responses, and then of course the cerebellum comes in the picture, and there is also a possibility of voluntary cortical control for these very late responses. And these responses will go all the way over the cortex, over the cortical pathway from visual to motor center to parietal cortex and so on. But we should not forget that there are these other centers like the superior colliculus, the tectum, the cerebellum, lower down, that are also important in some of the responses. So this is the end of this lecture, and I'd like to thank the CMPQ for providing me the help for having this visitor professorship that ended up with these papers that I illustrated. And I also like to thank my co-workers, of course. There's quite a few, there's not even a complete list. I've been, over the years, I've had the opportunity to work with very good students, and some of them are listed on this here. Thank you very much.
So we have time for questions. If someone wants to ask questions in Portuguese, I can translate to the professor. So. Jacques, I'm curious about how you are explaining this learning effect of reactive movements that is dealt with many people as some automatic and rigid and unchangeable responses, and you showed that they are improved by practice. How you are explaining this effect, this learning effect?
We have not looked at this in detail ourselves, but in this group that I work together with in Amsterdam, Miriam Panna also has done some studies some years prior to that. And she has shown that in the case where you have the stumbling, the tripping, that people adjust the way they walk. Of course, these are difficult experiments because after a while people know that this is the area where something can happen. And so even if you repeat it with trials that nothing is happening, there still is always the knowledge in these people that yes, it may happen, it may occur again. And so they have an adjustment apparently, and these adjustments have been described by Miriam Pineapples for the stumbling reaction. We have not done the same analysis for the experiment we did with the forbidden area, so we cannot really answer, but probably it will be similar to what Pineapples has described for the stumbling. And for the learning of the last part, the how to fall judo, I'd like to emphasize that this training is not so difficult. In half an hour time you can learn elderly to fall properly, but it is absolutely essential to repeat it, because we have also done a study later on to see what happens when you have the same people invited to the lab 1 year later, but they still would profit from the training, and the results were not so positive for some of them. The best results came from the elderly that fall regularly, because they fall regularly at home, and they have been using these techniques very regularly, and then it persists. But if they are not fallers and they are very infrequent, or they fall very infrequently, very rarely, then of course there's not enough learning so that they can maintain the level of proper fall. And in the most extreme case, the judo participants that we had, they could not fall otherwise than safely. So it depends on practice.
More questions?
Bom dia. Eu tenho estudado estimulação cerebral não invasiva, e o senhor falou sobre optogenetics. Gostaria de saber se ele já fez alguma coisa ou tem interesse com técnicas de estimulação cerebral não invasiva.
TMS?
Isso, TMS ou TDCS, que seria, TMS é a magnética e a TDCS é corrente direta. Se ele tem interesse em fazer alguma coisa, você já fez.
Além do optogenetics, se ele já fez, algum interesse, o que ele acha da técnica, das técnicas não invasivas?
The question is related to non-invasive techniques for brain stimulation, like TDS or electrical stimulation. If you have any information about this.
Yeah. Thank you. Well, TMS we have not used in this context. We have done some recordings of cortical activity using EEG and using NIRS, but we have not used TMS. I want to make a point though that there is a lot of studies these days on TMS and motor control. And that's fine, especially for the arm movements. There's some interesting data coming out of it. But for locomotion, not all that much interesting new data came out, and I try to explain why, because the cortex is relatively not doing much in gait. It's important for the precise placement of the foot, but for the whole process of locomotion it's not so important. And people that look at our field of motor control, they are, in my mind, a little bit misled by the fact that there's so much emphasis on methods like TMS, because then they start to believe, oh, it must be the cortex that's doing everything, because it's just a method that has enabled to do these studies, but it's not reflecting the importance in all these behaviors, especially not in locomotion. It's not because so many people are doing TMS that TMS, the cortex is important for gait, or it's not the most important part for gait. But the recording techniques are really very interesting these days, because in the last 10 years or so, there are now techniques possible to record during gait, and we've done some studies with the EEG, for example, and that's very interesting to show that there is even a phase-dependent modulation of the cortical activity during gait. And the reason that it works is that now we have very good filtering techniques, because with EEG, of course, you have the movement artifact, and you have to find ways to eliminate these artifacts, and these computer algorithms are now available to make that sort of filtering. So there are plenty of papers these days on also EEG and on NIRS during gait.
Bom dia, primeiramente obrigada pela fala, foi muito interessante. Eu tenho trabalhado com andar patológico de crianças com paralisia cerebral e atualmente eu faço com andar hemiparético, pessoas que sofreram AVE. E aí eu queria saber a ideia dele sobre o central gerador de padrão em relação a esse andar, ao andar hemiparético, se eles perdem tudo, se ele acha que é capaz de recuperar, se há uma reabilitação, se a gente tem como treinar o gerador central de padrão para a locomoção com essas pessoas. Quais são as ideias dele em relação a isso? Obrigada.
Posso não?
Elisa.
Melissa's question is about hemiparetic gait from stroke or cerebral palsy. And the question is if it can be the relation, the application of this module, the CPG, how it would could be improved or associated with this asymmetric pattern of gait.
Yes. Thank you very much for the question. Yeah, we've done quite some studies also on stroke. And we found that in the stroke gait there are these basic differences, implying that cortex has a role, of course, especially in fine positioning. We know, of course, that the slapping of the foot occurs in stroke patients. But we have concentrated more on these actions that I described that were believed from the animal literature, that is obstacle avoidance and precision stepping. And we found in the study that we did on the stroke that of course they were much worse in making this obstacle avoidance. But one of the interesting things that we discovered there in that study was that actually both sides were having difficulty, because we anticipated, of course, the hemiparetic side will be the one that has difficulty in. But that's not what came out, because, of course, if the hemiparetic side has to support while the other leg is just as important, just as difficult. So both sides showed the deficit, because the loading on the limb. The other thing, about the asymmetry of the CPG, I've not given that.
Yeah, because you have an asymmetric pattern, and how the CPG would deal with the situation, the symmetry between both sides.
Yeah, it needs more support because of the insecurity and the difficulty of gait. So what we found is that when we analyzed in detail the gait pattern of the stroke, there was a clear effect on the double stance phase. The double stance phase is, of course, the most stable of the two. And so there was a selective increase in the amount of time spent in double stance phase. Is that answering your?
Yeah. Actually, I have some issue more, because I wonder if the hemiparetic, especially, if they are able to learn the CPG again after the damage. I don't know if you have some thoughts about that, because The main objective for the healthcare or the professional to rehabilitation for these people is like a recovery the gait again. So it's possible to learn again the CPG, because the CPG is like we already have, but we are healthy subjects.
Yeah.
So
very interesting question, and in fact we have done some work. Let me first say that at the beginning we did not with stroke patients, but with spinal cord injured patients. So once we had made a statement, yes, there must be a CPG in humans, and implied also that it has this feedback loops of the afferents, the logical conclusion was that in these patients it should be possible to have some recovery of stepping if you could reanimate the CPGs in these subjects. And so we did a study with spinal cord injured patients that received no more rehabilitation. They were 6 years after the incident and they received no more treatment. And we had them in a very intensive treadmill training program and could show that this improved the gait. Even so many years after the lesion, it was still possible to train them under some conditions. And the conditions were that the appropriate feedback was given. So the trick is that you have to really control well the loading of these subjects. It's like the baby walking. If you do not support the baby, it's not going to step. And the same is true for this spinal cord injured. If they were too much loaded, they could not step. So it was very critical to have the appropriate amount of loading achieved. And then the second part is that they had to receive the afferent input that I just described is so essential to keep the process going. So that is achieved by having them on the treadmill, because the treadmill provides loading at the beginning of the stance phase, the unloading at the end of the stance phase, so that the swing phase can be generated, because there's no longer the inhibition from the load receptors. So you need to have a combination of these two elements. The support has to be right, and you have to provide the right afferent feedback. I described too the load, but I also briefly mentioned the importance of the flexors of the hip, the iliopsoas. And so, of course, again, the treadmill provides that, because with the treadmill there is a passive movement of the leg, putting it in full extension so that you stretch the iliopsoas and you help in the initiation of the swing phase. So using these elements, you can have success with the spinal cord injured. And of course, we are not the only ones that have been shown this. There are several groups in the world now that have had very nice papers and results by training with treadmill. And these days they also use exoskeletons and locomotor. And more or less the same applies for the stroke, because in stroke you also have the same problem, that the spinal cord does not receive the correct supraspinal input from the cortex. So it has to then rely more on, or learn to rely more on this afferent feedback. And so also in cerebral palsy, there are many studies that have shown that it can be helpful to have treadmill training, because it can provide the CPGs in the cerebral palsy and in the stroke patients with the appropriate input, so that you can reactivate as much as possible the spinal generators.
Okay,
Does that answer you?
Yeah. Thank you.
A point that is currently interesting our group is the combination of very fast reactions, like the tripping reactions you showed, and the flexibility in the response to, after tripping, for example, in your example, in your results, modulating the length of the step to avoid the prohibited area. How do you see this combination between very fast responses, faster than the involuntary movements, and flexible responses? What are the structures, neurostructures allowing for this?
Yeah, well, there's some literature showing that the expectation of what is going to happen can have an effect on the short latency responses as well. So the context is very important, and the level of expectancy, what you expect that could happen, that it influences the gain of these reflexes. So that is what people have found, and what I also believe that it is really indirect. So there is a difference between what you see in the very first trial, this response, and the responses that follow after, because after the first trial, you expect that something can happen, and this influences your reflex pathways, even the ones with short latency that are at the level of the spinal cord and the brainstem. I want to add one more thing, because we talked about pathology that may be interesting for some of the people that are working with Parkinson. That has particular interest now for me. And that is that the system that I described, this emotional locomotor system, can be relatively intact in Parkinson patients. And there is some very spectacular data about this, and you probably know the example, because these people are, as you know, when they are at home, they're very akinetic and make these very shuffling small steps. But if there is a fire in the house, they will be the first to run out of the house. And my belief is that this emotional locomotor system with the amygdala is the reason for that. So there are these different locomotor systems, and they cannot activate these various systems like the cognitive pathway, but they are still having the emotional pathway, so that they can, because many people do not understand how it's possible that you can have Parkinson people running away, but this may be the explanation. You will answer?
Tem professor, professor. Tem vários estudos recentes que vem mostrando muito, né, em pacientes com Parkinson, essa relação do sistema límbico, córtex pré-motor e a marcha, né, os sintomas motores. Até o freezing, ele vem sendo bastante explorado nessa parte. Então, a amígdala, esse medo de às vezes cair, o medo da queda, a ansiedade, o quanto afeta esses pacientes. Então, cada vez mais tem mostrado o quanto pacientes, freezing, não sei se todos conhecem, é um dos sintomas do Parkinson, que é um bloqueio da marcha. E os pacientes relatam muito o que acontece quando tem mudanças na direção da marcha. Então, a gente fala quando sai um pouco do automático, né, quando eles precisam virar ou passar por obstáculos, então eles congelam. Ou eles relatam também em momentos que eles estão muito ansiosos, né, ou preocupados ou com medo. Então, cada vez mais tem mostrado essa relação do sistema límbico, quanto afeta áreas pré-motoras, então de planejamentos, que estão relacionadas também à marcha, nessa parte cortical, e o quanto tem essa relação do sistema límbico, córtex pré-motor e pré-frontal e essas alterações da marcha que acontecem nesses pacientes. Mas ainda é bem recente, ainda não tem sido, não temos tanta explicação, mas realmente parece haver uma ligação entre essas áreas.
Alguma questão em particular ou você quer que ele comente mais sobre isso? More comments?
No.
Carol is commenting about exactly about this point of the connection between the limbic system, premotor areas, and the locomotor control, and the association with special events, making them anxious. Like I know that I'm going to be tested, so I become anxious about this. And she notes that it changes completely the behavior in these situations. So she made a comment. If you have further comments about this relationship between emotion and behavior in Parkinson's disease.
Yeah, I think that is a particularly interesting point, that is the relation with the emotional system. And as you described in Parkinson's patients, it's very well known that the freezing, for example, is linked to emotional state. And I had not the occasion to describe everything that is new, but one of the things that I would have added if I had more time, that is that for the locomotor system, it's of course important to have a start system. You have to start going slow or fast. That's the start system, and that's what I described. But of course, you also have to stop somehow. And I didn't describe that, but I did explain that in the beginning in my experiments with stimulation of the MLR, sometimes it didn't work. And I do know now why, because in that same structure in the MLR, there is also a population of cells that use GABA, and these are inhibitory systems. So you have a mixture of different locomotive systems, but also stopping system. You have a system to stop locomotion, and that uses other transmitter like GABA, and also different sites. With the optogenetics, it's now known that you can stop the locomotion by having certain sites above the MLR and also below the MLR in the reticular formation in the lower brainstem. So there are several studies now pointing out that there is a cascade of commands to stop the locomotion. You can go it from the basal ganglia to the MLR to the reticular formation. In all these different locations, there are cells that you can activate with light, with optogenetics, to make the animal stop. So we know a lot more about the stop system. And in relation to what you described for the freezing, for example, I think more and more people will realize that the freezing is probably this stopping system that is activated inadvertently. We all need the stop system because we all have to stop at one point our gait when we walk to a certain distance. But of course, it's a difference to have that stop system triggered inappropriately for emotional triggers that are not important for us. For us, passing through the doorway is not a scary thing. For them it is, and it makes the stop system work. But it is interesting to see, I think, that the organization is very similar for starting and for stopping. It's also a cascade, it's a sort of a hierarchy. From one is the pulse and gives a command to the next stage, to the next stage. It's over these different levels, spread over these different levels.
Well, we have studied some special cases, and there is a famous one about Parkinson's, still about Parkinson's, about some people have a great difficulty to walk because of freezing, but when biking they are very, very good, no symptoms of freezing. We assessed some special cases also. One in special, a case of a girl who had dystonia. She had very uncontrollable movements, but when skating, it disappeared. Do you have any idea about what are the circuits that could be evoked to explain these cases?
Yeah, it's extremely nice to see these videos where you indeed have these Parkinson patients. Prof has shown this video at Anke Snijders. And I do have some idea about it, because I think in the cycling, what is especially different is that it's a very different loading situation. In the usual gait, you have to support the full body weight in your stance phase, certainly in the single stance phase. In cycling, that's not the case, because the push period is in conjunction with the release period on the other side, and you don't have the same loads. And I think in the case of gait, that the loads feedback is deficient in Parkinson patients. There's some evidence from several groups that show that in Parkinson there are proprioceptive deficits, especially with the loading, perception of how much the limb is loaded. And I think this may be the clue of this difference between these various activities. The other thing is that there is also an automaticity in the cycling, because the start, when you have to start pushing for the next cycle, is given you because of the inertia of the flying wheel. So you have automatic cues that are used, which are not automatically present when you make steps. In fact, that's what we were discussing yesterday. If you want to look at example what in normal walking would be the equivalent of these cues, you need the stripes as they use in their house at regular intervals to have a cue to make the step, and that helps. And in cycling, it's automatic because you don't need the stripe. The cycling itself, the inertia, provides you with the trigger for the next push. That's what I think may be the background.
More questions? So, Professor Duysens, thank you very much for your lecture. It was quite enlightening. I believe that everybody enjoyed very much your talk today. Thank you. Thank you very much.